Hi, welcome to Unacademy. I'm Dr. Shivangi Shukla, your educator for biochemistry. Welcome all of you to this session. We are meeting after a long break, isn't it? Let me just check if I'm visible and audible to everyone. I'll just see if the live stream has started or not. Yeah, okay. All right. So let us begin. Today, as discussed earlier, I'm because we had taken long break. I had taken a long break. So to ease out the inertia slowly, I'm taking up small topics, interactive topics, which are not very drab and monotonous, not heavy on the theory part. Okay, so these topics will be like image of the day, mnemonics, a lot of mnemonics based sessions, and a lot of clinical application based sessions. And side by side, simultaneously, I'll be taking MCQ revision classes on the Unacademy app. All right. So let us begin. All right. As all of you would be knowing, who are following me here on this channel, I'm Dr. Shivangi Shukla. I've passed out from LHMC and did post graduation from MAMC. Yeah, so before the others join in, I'll just quickly, while the others are joining in, I'll just quickly take all of you through the different types of subscriptions available on Unacademy, and one very important announcement which I hope about which all of you would be knowing and you would be utilizing that. So the NEET PG subscription available on Unacademy is of two types, Iconic as well as Plus. Now, both of these will give you access to top educators from across the country in the field of medicine, and they will be quizzing you, they will be giving you theory classes, they'll be covering the entire syllabus of medicine and the other different subjects in medicine, medical field. And also along with that, they will be providing you with practice questions, mock tests, the printed notes, and all of this will be available in a simulated classroom environment, albeit online, but you will it will give you an even richer feel compared to the physical classroom. Okay, and the best part is the USP is that you can access these while on the go. Okay, even at the comfort of your homes. Okay, these special classes are enriched with a lot of state of the art features. So these are the current prices for the subscription, the plus prices and the iconic prices. But as I already told you, I'm here with an amazing offer for all of you. Now, before the 12th, before the midnight strikes tonight, if you apply the code DRSHUKLA Dr. Shukla and take any of the subscriptions, you are going to unlock 20% discount. Yes, 20% discount you will get on the whole of the price. So it will be 1/5 of the price slashed off. Isn't it amazing? Such a huge discount. So please do avail this opportunity, because the prices are in to be rising soon. So do avail this opportunity, and do not forget to put in the educator code DRSHUKLA Dr. Shukla while checking out, while buying the subscription. And remember, it lasts only till tonight. So please hurry up, guys. All right, and it is a win-win, because you are not here to lose anything. It is compared to the prices of other coaching institutes, these are peanuts. Okay, this is very less. You would be spending more on food in a particular month, in just 1 month, compared to the annual about 6 monthly subscription that they're offering after this 20% discount. Okay, so please do consider this. Now, so without further ado, I'll just quickly begin with the clinical applications part for today. Okay, Now, what am I considering? I am considering nucleic acids, okay, for clinical. Now, clinical applications, we have already completed around 3 lectures, okay, I think 3, if I'm not wrong. Insulin, in the first one, we discussed diabetes and then the biochemistry of insulin. And simultaneously, we also discussed collagen and PTH because they have more or less relatable structures, not exactly similar, but the disulfide linkages and the like. So we discussed that together. Then we discussed LFTs, then we discussed TFTs in the next session. So today we'll be discussing nucleic acids. But because it is an applied class, so I'll be taking the application part of nucleic acids. That is 2 things I'll be discussing. One is RT-PCR and the other is CRISPR-Cas technology. Now, both of these are very, very much in vogue these days, and being a student of biochemistry, you guys should be thorough with this. All right, so let us begin. I'll just give you a brief about what we are going to study. Nucleic acid, clinical applications, okay. So there'll be a lot of clinical application based classes for nucleic acids because regarding repair mechanisms also we'll have to take one class, okay, application based class. So it is very, very important. This one is the first in the league. Today what we'll be discussing is, as I've already mentioned, RT-PCR and CRISPR-Cas technology. Understood? Both of these we'll be discussing. Okay. Now just to give you a brief. Okay. Now regarding DNA synthesis, okay, let us start with DNA synthesis because that is what we are going to be focusing majorly on. Okay. So DNA is a double helix, right? It is a double helix. And DNA replication is what? Synthesis or replication. DNA replication is one very important keyword is it is semi-conservative. That is, one strand is conserved. Isn't it? I'll explain each and every term. It is a double helix, anti-parallel. Both the strands are anti-parallel. Anti-parallel strands. These are the keywords. So you should be in the habit of making notes while only taking down keywords. Okay, your notes should not be too much too verbose. They should not include a lot of words, a lot of paraphrases and all. They should only be point wise. Okay, so that will help you in reducing the bulk of your notes. That will help you in reducing the inertia to start revising the notes, because once you have very bulky notes, you tend to get really very apprehensive or lazy to start revising them. Okay, so do not make bulky notes. Keep true to the meaning of the word notes, and the note should only be the summary part. Okay, the summary of what you've read. All right, otherwise there's no use in making notes, right? Notes, if your notes are similar to the elaborate books that you're reading, there's no use making notes, isn't it? Anyway, so DNA synthesis or replication is what are the main keywords? Double helical structure, semi-conservative nature of replication, and the 2 strands are anti-parallel. Very important. Okay, now I'll make the structure of DNA something which every one of you knows. Now, so this is what? This is a double helix. This is one helix. Remember, if you guys have followed the MCQ classes on Unacademy, this is one alpha helix. Helix is helical structure. This is one alpha helix. So this is one alpha helix, and if you have a winding of another one, it will be a double helix. Similarly here. Okay, so where have you heard of a triple helix? Is there any molecule which you have read about which has a triple helical structure? Any one of you? A molecule with triple helical structure? Alright, it is a homework for you guys. We have discussed this time and again. Even at the start of this class, I mentioned about that. So if you guys can guess, if you guys can take a guess, it is a triple helical molecule, and one clue I'm giving you is that the handedness of the individual helices is opposite compared to the handedness of the overall structure. So opposite handedness. Now, if anyone can tell me in the comment section, or if you watch this lecture later on, then please do comment in the comment section about what I'm asking. So this is a homework for all of you. Triple helical structure is of which compound has a triple helical structure, which molecule? Anyway, so these are the two strands. Now, if this is running in the 5' to 3' direction, so this one will be running in the 5' to 3' direction like this. So this is anti-parallel. Now, anti-parallel, again, where had we read it? We had read it in the beta pleated sheets, right? The beta pleated sheets, if they are anti-parallel, then the hydrogen bonds, the interplate hydrogen bonds are almost collinear, and that is why they are more stable. Okay, and right-handed alpha helices are more stabler. Okay, so anti-parallel means the polarity of one, only polar chains can be parallel or anti-parallel. If something is non-polar, then there is no point of there being any direction, there being in any direction. So now this is running from 5' to 3'. So its 5' end is in close proximity with the 3' end of the sister chain. Okay, so this is what is anti-parallel. So double helix, anti-parallel, and what is semi-conservative nature? Now between these two strands there is this hydrogen bond, and the hydrogen bond is existing between the nitrogenous bases of the individual of the strands, isn't it? These strands have nitrogenous bases which are linked through the hydrogen bond. Okay, now for DNA replication, what happens is these two strands must separate. Okay, there is denaturation of DNA. Denaturation of DNA means separation of the two strands. That is separation of the two strands. So now by common sense, can you guys tell me which bonds will be broken during denaturation? Can you guys tell me during denaturation of DNA, which bonds will be broken? Which bonds will need to be broken to separate the two strands? Very important. That is what we discussed here in the last slide. These are the rungs of the ladder. Rungs means the steps of the ladder. These are the hydrogen bonds between the nitrogenous bases. They are what? They are the rungs of this ladder. Now, to separate these two strands, you have to break these rungs. So, denaturation of DNA or separation of the two strands of the DNA involves destruction of hydrogen bonds. Very important. Ask your question. So see, most of the people got confused in this. So this is a very basic question. Whenever we start reading, this is the first thing that we read, that the two strands must separate. And how do they separate? By dissolution of the hydrogen bond, breaking of the hydrogen bond. So this is what is denaturation. So these small keywords, you should always make sure that the keywords that you are using in common parlance, you know the meaning of those keywords. Okay, it is not just because that is what you are quizzed on. Everyone who is preparing knows important concepts. Everyone can learn the facts and figures, but only those who know what they have learned, only those who understand the concepts will be able to become toppers or will be able to clear by a huge margin. Okay, So always understand and then learn. So now what happens is, since these two strands will be separated, okay? Now these two strands have separated. Now what will happen? Now DNA polymerase is an enzyme. DNA polymerase is an enzyme that is going to add new nucleotides that will be complementary to these, okay? It will add new nucleotides that will be complementary to these. And these are what? These are the two daughter DNA that have formed. Now, how is it so? It is not so simple. I'm going to tell you. Okay. Now, imagine this replication fork. Okay. Now, this is the 5' to 3' end. And this here is the 5' to 3' end. Okay. Now, these already have the hydrogen bonds intact. And here what has happened is the hydrogen bonds have separated. Okay. Now what will happen is, hi Nitish, how are you? What are you preparing for? Are you a first Prof student or are you appearing for NEET PG entrances? How is your preparation going on? So now what will happen? See, going back to our lecture on DNA synthesis and DNA replication, this 5' to 3' end, what happens is, the synthesis DNA polymerase enzyme works in the 5' to 3' end only. So it will start synthesizing from here. Okay, it will start synthesizing. This is the DNA polymerase enzyme. It will start synthesizing the daughter strand in this direction. So this is 5' to 3' direction. Okay, understood? And this is continuous synthesis is occurring. But what will happen here? Here, as and when the replication fork opens, there will be short segments that will be synthesized. And these segments are called what? They are called Okazaki segments, fragments. They are called Okazaki fragments. And also, to prevent these two strands from sticking together, what do we have here? We have, to prevent these two strands from sticking together, we have SSBs. What are SSBs? Come on, tell me guys, what are SSBs? And here, there is a complex that is the primase complex. Okay, sorry, the primosome complex. Sorry, not the primase. The primosome complex. Okay. Now there is another homework for you guys. To tell me the primosome complex is made up of two different enzymes. What are these enzymes? You have to tell me. Okay, so there are different categories of enzymes involved in DNA replication. One is topoisomerase. One is helicase. Helicase, as the name suggests, will break the helix, will open the helix. Topoisomerase will relieve the strain. Single strand binding proteins. SSBPs, sorry. Primase enzyme. Okay, so what are these enzymes? You have to tell me, okay? And of course, DNA polymerase enzyme. So these are the important enzymes that you guys have to answer in the comment section. Why? Because this is not a theory class, this is an application based class, and theory we have already read. Okay, so these small fragments are what we discussed. They are Okazaki fragments. Now they will be joined together by an enzyme that is DNA ligase. So many enzymes we have read of in DNA replication. Understood? Most important is DNA polymerase, about which we are going to read in RT-PCR. So understood, first the DNA strands they separate, then there is addition of new strand complementary to the original one, and then what happens is the daughter DNA then they form. Understood? Now look at this diagram for 1 minute and then we'll continue. Okay. Okay, understood? Now we'll start our discussion, that is RT-PCR. Okay. Now, first of all, the full form. Okay. RT is reverse transcription polymerase chain reaction. Okay, reverse transcription polymerase chain reaction. This is RT-PCR. All right. Now, each and every word we have to take note of. Okay. What is polymerase chain reaction and what is reverse transcription? So let us start with the first term. Okay. Reverse transcription. Now, what is transcription? First of all, transcription is conversion of DNA to RNA, that is synthesis of mRNA from DNA. What is transcription? Synthesis of mRNA from DNA is transcription. Now, then what will be reverse transcription? It will be synthesis of DNA from RNA. So, transcription is RNA from DNA. But reverse transcription is DNA from RNA. So, this is reverse of what is occurring in nature. In nature, what happens is DNA transcribes into mRNA. But when reverse happens, what will happen? Hi Atharv, how are you? But when reverse happens, what is it called? It is called reverse transcription. Now, where does it occur? It is not very common. In some viruses, which are retroviruses. Okay, retro means reverse, retrograde. So, as the name suggests, their genetic machinery is working in the reverse direction. What happens is they will obviously be RNA viruses. These are RNA viruses which convert RNA into DNA using the mechanism of reverse transcription. Now, how does it occur? It is a three-step phenomena. We'll discuss this. We'll discuss reverse transcription here. Okay. Now, the first step is where, now suppose we are starting with a single-stranded RNA virus. Okay, so this virus contains a single strand of RNA. Now, this single-stranded RNA will convert to a hybrid of RNA and DNA. How? Using the enzyme RNA dependent DNA polymerase. Now, is it very difficult? Hey, thanks Athar. So does this seem to be difficult to you? RNA dependent DNA polymerase, does this seem to be confusing? But I'll help you revise this. I'll help you learn this. This virus contains single stranded RNA. This virus contains RNA. So whatever enzymes it will have, it will be, those enzymes will be dependent upon the RNA. They will function with RNA as the base. And what are they forming? They are forming DNA. So what is this polymerase? What is polymerase? Polymerase is something that synthesizes, that polymerizes. So DNA polymerase is something that synthesizes DNA. RNA polymerase is something that synthesizes RNA. So here what is getting synthesized? DNA. So what will be the polymerase? DNA polymerase. So there is this polymerase enzyme that is dependent on RNA and that is synthesizing DNA. And this is what is happening in retrovirus. This is what is happening in reverse transcription. So a shorter form for this enzyme is also, what is this enzyme also called? What is this enzyme called? RNA dependent DNA polymerase enzyme is also called reverse transcriptase enzyme. Understood this? Because this is what is causing reverse transcription. Okay, so the first step of reverse transcription is formation of a hybrid, RNA DNA hybrid. Now this retrovirus contained single stranded RNA. Now this single stranded RNA now has one, starting off it had just this single stranded RNA. Now after the action of this reverse transcriptase or RNA dependent DNA polymerase, what has formed is a hybrid of RNA and DNA. Now this molecule is so funny, isn't it? It's weird. It contains one strand of RNA and the other strand of DNA. Now what will happen? Now we want, because our main objective is to convert this single stranded RNA to a double stranded DNA. So now we want to get rid of the RNA. So there is this another enzyme, that is RNase that will remove the RNA strand. Then what will be left? Just one strand of DNA. Okay, then what will happen? It will remove the RNA strand, so leaving behind only one strand of DNA. Now, starting off, we had one strand of RNA. Then we had a hybrid of RNA DNA. Now, after the second step, we had just one strand of DNA. Okay, now in the third step, what will happen is, in the third step, there will be DNA replication. Now, this single strand will then replicate to produce another strand using DNA polymerase. Understood? So, this is how reverse transcription occurs. First step is reverse transcriptase, second RNase, and third step is DNA replication. So this is how a single stranded RNA converts to a double stranded DNA. Okay. Now this is what we are going to apply in reverse transcription. RT-PCR. What are we going to do here? Suppose, okay, so now let us talk about the most common, most talked about virus. Okay, that is the COVID virus. Okay. COVID-19 is the disease and the name of the virus is what? The name of the virus is SARS-CoV. SARS novel coronavirus. Okay, this is the agent or this is the virus. Okay, now the SARS-CoV, it is a single stranded RNA virus. Okay, it is a single stranded RNA virus. Now what happens is, suppose we have to conduct an RT-PCR test on someone who is a suspect of COVID. Alright, so what will we do? what are we going to do? First of all, we'll take the sample. Okay, now we'll collect the sample. You know, oropharyngeal swabs and nasopharyngeal swabs are taken. They're transported in a carrier medium, and then they are tested in the lab. Now, inside the lab, what happens is you take out the sample, and first of all, the first step is reverse transcription, because we know that the sample has RNA. If at all the sample has the SARS-CoV virus is present, then that will have RNA. So that RNA, for that to detect, we have to first convert it into DNA. Okay, so the first step is reverse transcription. Now we are going to conduct reverse transcription on the sample. So the single-stranded RNA has then converted to double-stranded DNA. Alright. Now the second step is PCR, or polymerase chain reaction. And this is what we are going to study here. What happens here is, suppose this is a double-stranded DNA. Okay. Now this is a double-stranded DNA molecule. Now what will happen is, we will subject it, the helicase enzyme. What does the helicase enzyme do inside our body? It separates the two strands. Now what we want is, outside the body, we want to conduct the same procedure. Inside our body, how DNA replication occurs, we have discussed. So now outside the body also, we need to replicate the same procedure. So we won't have the same enzymes, but we'll have some similar mechanisms. So these two strands can be denatured using either enzymes or high temperature also. Similar to any proteins, denaturation of proteins occurs at high temperatures also, using it can occur through chemical, thermal, biological, whatever means. So using high temperature at around 1000 degrees Celsius, what will happen? These two strands will separate. But you remember, inside the body, what did we have? We had single-stranded binding proteins that were preventing the reattachment or the resealing of the two strands, because they have complementary nitrogenous bases which are attracted towards each other to again form the hydrogen bond. Okay, so therefore inside our body, whenever there was a replication formation by separation of the two strands, then there was a tendency for the two strands to come together. Therefore, they were separated by the single-stranded binding proteins. Similarly, here, till the time the temperature is high, these two strands will be separated. Okay, because the hydrogen bonds cannot form at such high temperature. The enzymes are inactive. But once you lower the temperature for the enzymes to act, because now that you have separated the two strands, you will want replication of the two strands. How will replication occur? By addition of DNA polymerase. But that DNA polymerase cannot function at such high temperatures. Okay. So once you lower the temperature for DNA polymerase to act, these two strands are going to come together. So what will you do? We want a special type of DNA polymerase that can function at very high temperature. Okay, and what is that DNA polymerase called? That DNA polymerase is called, that special type of DNA polymerase that can function at very high temperatures, that is called Taq polymerase. What is it called? Taq polymerase enzyme. Okay, it can function at very high temperatures. Okay, now this Taq polymerase enzyme, what will it do? It will start synthesizing it will start synthesizing the daughter DNA strands. So this one will have one daughter strand, and this one will also have a complementary daughter strand. And then what will happen? We have two daughter DNA from the first one. What is special about Taq polymerase? What is Taq? Taq is thermophilus Phylus aquaticus. That is, this is an organism that survives at very high temperatures. Okay, this is an aquatic organism that is a thermophile that loves high temperatures, that can survive at very high temperatures. So the DNA polymerase that is present in this organism is able to function at high temperatures. So this is what we have extracted and we are utilizing in vitro. Okay, so two strands have been produced. Similarly, there will be repetitive such cycles so that a small amount of viral RNA, which has converted to DNA, a small amount of viral nucleic acid can be magnified, amplified. If this cycle is run for hundreds of times, not hundreds of times, if it is run for tens and 20 cycles, if this is run, then a small amount of DNA will be concentrated and it will be amplified. So this is what is the keyword? Amplification. Okay, and then what happens is you add DNA probes, complementary probes, and if they attach to the viral, if they attach to particular areas, then you know that this is what is the COVID virus. Okay, preformed DNA probes, if you add to the sample and there is fluorescence, how will there be fluorescence? If those probes attach to particular areas, then that means that the sample is positive for COVID virus. Alright, so I think the time is up for the session. We have an upcoming open house session as well at 6:35. So it's already time for the open house session. So I'll just stop here. And the applied concepts of RT-PCR, there are a lot of concepts left still. And CRISPR-Cas technology will be taking up tomorrow or day after tomorrow in another clinical applications session. Okay guys, then see you. Bye-bye. Join me in the open house session. Go download the Unacademy app if you haven't till now. And put in the educator code DRSHUKLA, Dr. Shukla. If it asks you for an invite code, you can put in DRSHUKLA, Dr. Shukla. And go and follow me there in the NEET PG category, Shivangi Shukla. And there you will find my open house session that is on Ask Me Anything. So the session is active now. I'm going to take the session. So kindly join the session. Ask me anything, any doubt regarding your preparation, you can ask me. Open house session on the Unacademy app, NEET PG category, Dr. Shivangi Shukla. You go and check. The session is live now. Alright guys. Thank you. Bye-bye. And also join me at 7 p.m. on the image-based session once. Image of the day. That is for tonight. Okay. At 7 p.m. sharp. So back-to-back classes are there. That is why I'm rushing. See you guys in the open house session. Bye-bye. If there's any doubt regarding this session also, you can go and ask me there. Okay. Unacademy app. NEET PG category, Shivangi Shukla. Go, there'll be a blue screen for the open house session. You can click on that and you'll join that. Okay. So 1:1 audio interaction we can have there. Alright. Bye-bye. Thank you.